This protocol fills a technical gap for the biochemical study of C. elegans and expands their usefulness as a model organism. This technique is both simple and robust, allowing for consistent isolation of functionally active C. elegans nuclear extract.
Begin by placing the synchronized L1 animals on 10 150mm nematode growth media containing plates seeded with Escherichia coli OP50, and allow the animals to grow for 48 hours at 20 degrees Celsius until they reach the L4 stage. Once the hypotonic and hypertonic buffers are prepared, clean the Balch homogenizer by flooding the grinding chamber with 70% ethanol. Then rinse the chamber with deionized water to remove the excess ethanol. Insert the tungsten carbide ball into the grinding chamber. Cap the ends of the Balch homogenizer's barrel, and secure the caps with the provided thumb screws. Then, prepare 5 ml of the complete hypotonic and hypertonic buffers per sample as described in the text manuscript. Next, fill a sterile 2 ml syringe with 1 ml of complete hypotonic buffer and gently flush the grinding chamber of the Balch homogenizer, leaving approximately 500 microliters of complete hypotonic buffer in the chamber. Store the flushed homogenizer on ice and let it cool for 30 minutes. Collect the well-fed L4 animals with M9 buffer in a 15 ml conical tube, and centrifuge the animals at 1000 times G for 3 minutes. Remove the supernatant and continue washing the animal pellet until the supernatant